I see. So what was the mechanism of action that you guys discovered? Do they all have different mechanisms of action, all these different plants?
They do. There's three main mechanism of action to release stem cells that all have an impact on one aspects of stem cell function, and it's called the CXCR4 SDF1 axis, meaning that SDF1, stromal derived factor 1, is the compound that an injured tissue releases locally to trigger the migration of stem cells into that tissue through the expression of adhesion molecule. It is the exact same phenomenon that the bone marrow utilizes to maintain stem cells in the bone marrow and recapture them from the peripheral blood. So stem cells are naturally designed to home to the bone marrow and stay there. And when you disrupt that signal in the bone marrow, it makes them be released. So you can do that in various ways. The natural way that the body does is that your injured tissue will release granulocyte colony stimulating factor as one of the main mechanism for stem cell mobilization. When that compound reaches the bone marrow, it triggers the secretion of an enzyme that digests SDF1. So in the absence of SDF1, the bone marrow loses its ability to keep stem cells in the bone marrow, so they get entrained into the blood flow. So that is your natural method of mobilizing stem cells. You have drugs that have been developed, like AMD 3100, for example, that are blockers of CXCR4, which is the receptor for SDF1 on stem cells. You block the receptor, then the stem cell then no longer responds to SDF1, so they get into the circulation. So other mechanism of action is that some plants will fool your body, if you want, to start to produce granulocyte colony stimulating factor or stem cell factor, which is also a mobilizer. So, and the mechanism that we discovered with AFA is that, I need to go a little bit deeper in this. The receptor for SDF1 is not always on the membrane. It's a preformed receptor that is right under the membrane surface. And when the stem cells is activated to listen for SDF1, then it pops out of the membrane. So that mechanism of popping out takes place by the activation of an adhesion molecule called L-selectin. So what we thought is that the response that we had with AFA was much weaker than what we know of GCSF, for example. So we thought maybe that could be a mechanism of action, meaning we block L-selectin, so all the CXCR4 existing on stem cells are there, but we can no longer upregulate them. So all the stem cells that have fewer of these receptors then starts to shed them, and they get sensitive, if you want, to mobilization if you block L-selectin. So we fished for an L-selectin blocker in AFA, and we found that we have one. So what we did is we basically took recombinant L-selectin that we embedded into very, very small microscopic magnets, and then we incubated AFA extract, water extract of AFA with these magnetic beads. So we fished out any kind of L-selectin ligand, and then after that, you put a magnet on the side of the test tube, you wash everything, and then after that, you detach whatever was attached to these to the L-selectin, and we ran that through gel electrophoresis, and we got like very, very clear band. There is one very specific molecule which is suspected to be a glycoprotein that acts basically as an L-selectin blocker. So that's what we have in AFA, the blue-green algae. Other compounds have other benefits or other mechanism of action. Panax notogensin, for example, will do two things. To me, it's phenomenal that nature has this kind of effect in the body. It will trigger the release of stem cell factor, but will also reduce SDF1 in the bone marrow and increase it in the bloodstream. So you change the concentration gradient for the molecule that attracts stem cells. So stem cells suddenly get attracted to the bloodstream. So there are different mechanisms of action, but they all act through this sort of single mechanism that is called the CXCR4 SDF1 axis.